Hello everyone. In our today's video, we will be learning about an important biomolecule, which is nucleic acid. So as we know, nucleic acid is a naturally occurring chemical compound that is capable of being broken down to yield sugar and nitrogenous base and a mixture of a nitrogenous bases and phosphoric acid. So this nitrogenous basis, as later on when it was investigated, was found to be of two different derivatives, that is purine and pyrimidine. We'll be talking a detail about it, its composition, and how actually it forms a bond and how it goes for the formation of nucleic acid. Nucleic acids are the main information carrying, you can say, center or carrying molecule of the cell. Okay, and by the process of actually directing the process of protein synthesis in our body. Okay, there are two important types of nucleic acid, okay, or two types of nucleic acid which we have in our body, which we call as DNA and RNA. As all of us know that DNA is kind of a master blueprint, okay, for life and constitute the genetic material in all free living organisms, okay, and in most viruses. RNA, although it does not carry the genetic material, but it carries in some of the viruses. And not only that, but RNA is very important actually, and present in all living cells, where it carry out the important process of making of proteins. Okay, so that is what actually we know, the basics of nucleic acid. So in our today's video, we'll be trying to go more than this. We'll be trying to understand that how we can really take a base, a sugar, and a phosphate and bring them together and make a nucleic acid, and how that is possible, how this chain grows, and how it gives you DNA and RNA. What is the structure of DNA and RNA? That is what we'll be going to do in our today's topic. So let us go with that. So starting with the introduction, as we have already discussed, nucleic acid constitute an important class of biomolecules which are present in all the living cells, okay, nuclei of the living cells. They play an important role in the development and reproduction of living organism, as I have already explained it to you. They act as instruction manual, as I've told you, it's a master blueprint, okay, and not only for the genetic information, but also for protein synthesis, okay, and the genetic information are transferred with the help of that from parent cell to the new cell, okay, in the case of subdivision or cell division. Okay, the nucleic acids are biopolymer, very important, okay, the polymers which are present in the biological system, which are synthesized in the biological system. Okay, and since it is a polymer, every polymer will have its monomer. The basic question is, what is the monomer of a nucleic acid? So the monomer of the nucleic acid is a nucleotide. Okay, they are also referred to as polynucleotide. That's why, okay, it is made up of many nucleotide unit together. Okay, so that's why we call it polynucleotide. Nucleic acid are of two types. We know that is DNA and RNA. DNA, the full form is deoxyribonucleic acid, and RNA is ribonucleic acid. Okay, so let us try to go ahead with this and understand them in detail and about its chemical composition and its formation. So constituents of nucleic acid, as we have already discussed that there are three major chemical components which are present in nucleic acid: a sugar, a nitrogenous heterocyclic base, and a phosphate group. So which sugar is present, in what form it is present, what nitrogen base is present, and what is this phosphate group? Let us try to see it in detail. So let us first go with sugar. So two types of pentose sugar have been obtained in the hydrolysis of nucleic acid, and they are this two. Okay, the first one is beta D ribose, which is present in RNA only, and the another one is beta D 2-deoxyribose, which is present in DNA. We call it 2-deoxyribose because you can clearly see in the second position over here you have oxygen, over here you don't have. Okay, deoxy, removal of the oxygen, okay, from ribose gives you deoxyribose. Okay, this is the furanose form. Okay, we call it pentose sugar because it has 5 carbons. Okay, so coming for next, that is the nitrogen bases. Okay. So in all, there are 5 nitrogen bases, and they are heterocyclic nitrogenous base. And what we found out is that they are derivative of 2 nitrogen base, that is purine and pyrimidine. Okay. So this is purine and pyrimidine. Okay. Remember that purine and pyrimidine themselves are not present in the DNA and RNA, but their derivatives are present. So let us try to see what are those derivatives. First, let us try to see the structure of purine derivative, which is this 2, which we called as guanine and adenine. Okay, which is represented as G and A also. For pyrimidines, we have 3 types. Okay, the first one is thymine, and remember that thymine is present only in DNA, not in RNA. Then the next we have cytosine, which is present in both, and uracil, which is present only in RNA, not in DNA. So these 3 are the pyrimidine base. Okay, they are derivative of purines and pyrimidine. So in all, we have 5 nitrogen bases in DNA and RNA. And the final constituent chemical component which we have is the phosphate, which plays a very important role in linking the nucleotides, okay, as we have already discussed, which is the monomer of this biopolymer. It helps to link that together, okay, and that is what actually is this, okay, that is the phosphate group, so which you can see in the figure. So after learning that what are the various chemical composition, or where are the various chemical components of nucleic acid, let us try to go for the formation of this nucleic acid. So before going into nucleic acid, we must understand how nucleotides are formed. And before going for nucleotide, let us first try to see how nucleosides are formed. So what are nucleosides? These are base and sugar unit, okay, present in the nucleic acid chain. Okay, so basically when base and sugar, they combine together, what you get is a nucleoside, which you can see in the figure. The first figure is of ribonucleoside, because the sugar is ribose, and the another one is deoxyribonucleoside, because the sugar is deoxyribose. Okay, and one point you have to note over here, that the base will form bond with the sugar through the C1 carbon of the sugar, and the carbon of the sugar are represented by actually using prime, okay, so that we don't get confused with the numbering which we do in base also. So remember that we use primes in the numbering of the carbons, okay, in the sugar. Now, a very important thing over there, okay, to understand is the base will form bond with the C1 position of the carbon. And if your base is a derivative of pyrimidine, then it will form through the N1, okay, the first nitrogen atom, it will form bond with the C1 of the sugar. And if it is a purine, then the purine will form with C9, okay, or the 9th position, sorry, not the C9, but the 9th position of the purine with that particular C1 of the sugar. Okay, so remember that purine forms bond with the 9th position, and the pyrimidine forms bond from the 1st position. Okay, now coming for this nucleotides. Okay, so once we have understood what is a nucleoside, nucleotide is nothing but nucleoside plus phosphate. So if you add phosphate in a nucleoside, you get nucleotide, which means base plus sugar plus phosphate is a nucleotide, which you can see in the figure. Over here we have written X. That depends whether if it is H, it will be deoxyribose. If it is OH, it will be ribose. The base is in C1 position, and very important, the phosphate is linked to the C5 position. Okay, the carbon number 5 of the sugar, whether it is ribose or whether it is deoxyribose, and this linkage which actually it forms is known as phosphate ester linkage. Okay, or the phosphate ester, we call it, of the nucleoside. So it forms a phosphoester of the nucleoside, which is nothing but a nucleotide. Okay, so that is how a nucleotide forms. So this is basically the unit which gets repeated again and again to give you the whole nucleic acid. So now what we'll be trying to understand is how this unit again and again repeats to give you the long chain of that biopolymer. Okay, so how that happens, that is what we will be trying to understand now. Okay, so coming for the formation of nucleic acid or the primary structure of nucleic acid. Okay, so this is what basically happens. Okay, so remember that in this particular nucleic acid, okay, in this what I have shown is I have shown a dinucleotide. Okay, and it is very important to note that the C5, okay, which was bonded with the phosphate, combines with the C3, okay, carbon of another nucleotide. Okay, so the bond or the linkage through which the nucleic acid is formed from the nucleotide is through this 5'3' linkage. Okay, so the two carbons which are involved in two separate nucleic acid is nucleotide is in one nucleotide, the 5' carbon where the phosphate is linked, that phosphate gets linked with another carbon of another nucleotide through 3'. Okay, ultimately what we'll have is at the end, in one end we'll have a 5' carbon and another end we'll have a 3' carbon. Okay, so let us try to see that. Nucleic acid are polymers of nucleoside or nucleotides, what we know. Okay, during the formation of nucleic acid molecule, thousands of molecules of nucleoside or nucleotide undergoes polymerization with the elimination of water molecule. Okay, the nucleic acid backbone consists of alternating sugar phosphate residue. Okay, so the nucleic acid backbone, okay, the chain which is made up of will be made up of alternating sugar and the phosphate backbone, which I have shown you earlier. The sequence in which the 4 nitrogen bases are attached. Okay, so what will happen is the backbone will consist of the sugar, then phosphate, then sugar, then phosphate. This phosphate and the sugar will be bonded in the 5' and the 3' position by the elimination of water and making a phosphoester linkage. And after that, what happens is each nucleus or each sugar will be bonded with a base, and the sequence in this which the bases are attached to the sugar phosphate backbone is referred to as the primary structure of nucleic acid. So whenever anyone say what is the primary structure of nucleic acid, it is the sequence of this bases which are attached to the sugar phosphate backbone is nothing but the primary structure of nucleic acid, which I have shown you in the earlier picture or in the diagram. Now, let us come for the structure of DNA. Okay, what we know that the 3-dimensional structure of DNA was first studied by James Watson and Francis Crick, okay, in Cambridge in 1953. Okay, and remember that, and all of us, as we know, that they proposed a double helix structure for DNA. Remember that according to Watson and Crick, okay, according to Watson and Crick, DNA molecule consists of 2 right-handed helical polynucleotide strands. So this which you can see is a strand of that polynucleotide which we have just seen, that how it forms. No, so it runs from, as I've shown you, it runs from 3' to 5' end, and another one will run from 3' end to 5' end. Now I hope we can understand what is this 3' and 5'. It is the number or the carbon atom number in the sugar. Okay, and remember that this 2 right-handed helical polynucleotide called together in the form of a double helix, which you can see in the figure, around the same central axis. Okay, it will have the same central axis. The 2 strands are anti-parallel. You can see one of them is running from 5' to 3' phosphodiester linkage, and the another one runs from 3' to 5' phosphodiester linkage. Okay, and those bases, very important. Okay, you can see in the figure, we are writing the abbreviation of the bases: G guanine, C cytosine, T thymine, A adenine. All this actually bases, if you see in that particular molecule, what you'll find out is that they will be perpendicular actually to the plane, okay, or to the helical axis. You can clearly see, okay, will be facing one another, and this 2 strand will be held together by the hydrogen bond which this base will form. And very important, a particular base can form hydrogen bond with only a complementary base only, or with a particular base only. Like a guanine can form hydrogen bond with cytosine only, and a thymine can form hydrogen bond with adenine only. So if in one strand, if you'll have guanine, in another strand opposite to it, you'll always have cytosine. If you'll have thymine, you'll have adenine. If you'll have adenine, you'll have thymine. If you have cytosine, you'll have another side guanine. This is what we call as complementary base pairing. Okay. So let us try to see that in detail. So DNA molecule is a long and highly complex spiral twisted double helix ladder-like structure, which you can clearly see in the screen. The two polynucleotide chain or strand are linked up by hydrogen bonds, okay, what I have told you, between the nitrogen bases molecules, okay. So the nitrogen base molecules forms hydrogen bonding, okay. Adenine always links with thymine, that's what I've told you, okay. And with the help of two hydrogen bond, which I'll be trying to show you in our next slide, and the guanine, okay, with always cytosine. So one purine will be forming hydrogen bond with another pyrimidine, okay, and there will be three hydrogen bond between guanine and cytosine. We'll be trying to see that. Hence, the two strands extend in opposite direction, okay, and are anti-parallel and complementary. So very important, remember that the two strands are anti-parallel, they run opposite to one another, and always remember that they are complementary to one another, okay. The following fundamental relationship exists between the in the molecule. Thymine combines only with deoxyribose sugar and uracil. Please remember that. Only with ribose sugar, and uracil only with ribose sugar. Other bases combine with either of the two sugar. Okay. The sum of purines equal to the sum of pyrimidines always. The molar proportion of adenine equals to that of thymine. The molar proportion of guanine equals to that of cytosine. The double helix is 20 angstrom. So this double helix is 20 angstrom. It completes a spiral at every 10 nucleotide pairs at a length of 34 angstrom. That is very, very specific. Okay. So at every spiral, you will have 10 nucleotide and it will be having a length of 34 angstrom. Sequence of monomers, that is nucleotides, which I have already explained to you, may present innumerable variation. Evidently, innumerable varieties of DNA exist in organisms. So there will be innumerable, okay, infinite number of such sequences are possible, okay, and with such sequences, you will have innumerable numbers of DNA existing in organisms. Okay, so that is how we can understand the structure of DNA. Now, coming to that complementary base pairing, which we were focusing more on that, how actually they form hydrogen bonding. So this is how it forms hydrogen bond, okay, where I have highlighted it. You can see clearly that is the hydrogen bonding, okay, or the atoms involved in hydrogen bonding. This is thymine and this is adenine, and you can clearly see they form two hydrogen bonding over here. Okay, so this dotted lines are the hydrogen bond. Similarly, you will have cytosine and guanine, which you can see clearly they'll have three hydrogen bond. And it will always happen that if in one strand you have thymine, opposite to it will always be adenine. And if you have cytosine, opposite to it will always be guanine. That is what we call as complementary base pairing. So that is how we understand that how DNA, its double helix is holded actually by the hydrogen bonds. Okay, so if you try to analyze now, so every possible actually, or how those two helix are bonded to one another is through this hydrogen bonding of the base, and the base which are forming hydrogen bond are very specific and complementary to one another. It has to be thymine and adenine and cytosine and guanine, only opposite to one another in the two strands. Okay, so that is how we can understand the structure of DNA, structure of nucleic acid, okay, how actually the basic component combines together to give you the monomer unit, and monomer unit finally gives you the nucleic acid or the DNA, okay, what we have learned so far. So lastly, let us just try to understand what is the function of nucleic acid. We'll not be going into so much detail, but as we know that nucleic acid are very important and they perform two most very important functions, okay, in biological system. And the two important biological function is replication, okay, and protein synthesis. Okay, so these are the two most important function of nucleic acid in biological system. Okay, so that is all about nucleic acid and DNA and RNA, and what are the various chemical composition, how they combine together to give you nucleosides and nucleotide, and finally gives you nucleic acid, and what is the double helix structure of DNA and how they are holded, what do we mean by complementary base pairing, and finally what is the function of nucleic acid. So that is what actually we have learned in our today's class. So thank you everyone. Do subscribe for this channel for explanation on various topics in chemistry.